We're still in the mitochondria. So what happens in the mitochondria is, well, maybe going back to that previous slide, here's where I get to ask the question. How many carbons you got left? From your original 6, how many do you have left? The answer is 0. Because you took 6, split it apart into 3. You took the 3s, tore off, released CO2, okay, and you only had 2 left. You stuck them into Krebs cycle and you pulled off those 2, you got nothing. So you have no carbons left. Okay, now ask the question, how many ATP have you formed up till this point in time? So you've run out of carbons. They're gone. You're done. But how many ATP have you formed? Well, in glycolysis, you got net 2. And in Krebs cycle, because you went through twice, you got 2 more. So how many ATP are you sitting on? Okay, well, you're sitting on 4 total up till this point. But you've got no carbons left. So how are you supposed to get this whole 36 to 38 ATP if you've got no carbons left and you only have 4 ATPs at this point? Well, the answer is all that stuff you've been filling your pockets with. The NADH, the FADH2s, you've been filling your pockets with this stuff. And now's the time to go shopping because your store of choice only holds a sale once a year. Okay, or it's the big ski swap. Okay, you're going to buy your skis, save them for your car. You're going to make your big purchase. And what's the big purchase going to be? Big purchase is going to be 32 to 34 ATP. You already got 4. Okay, but now you want the bulk of them. And where we get those? We get those in the mitochondrium through electron transport. And basically what happens is, here's where my pockets that are full of these NADHs and full of these FADH2s that I've picked up those hydrogen ions and electrons. And this is where I'm going to basically go through this hot potato system of passing these electrons along a system of proteins where you take them, no, you take them, no, you take them. So I'm going to be releasing that energy and passing or transporting electrons for the goal of making all of those ATPs we want. So we're going to go through electron transport. And basically what it is it's a series of oxidation reduction reactions. Now I've introduced that once before, but as a reminder, so it'll make a little bit more sense. In that process, remember in a previous lecture, I talked about the mnemonic, I think it was module 2, oil rig, that an oxidation reaction is loss of electrons and a reduction is gain of electrons. So what's basically happening here is I'm going to strip electrons from one molecule to give to another, which then take them off that one to give them to another, and take them off that one to give them another. But through that process, I'm going to be releasing energy, and I'm going to release energy to run hydrogen ion pumps. So active transport, to move hydrogen ions to a given place, to build them up. And so basically kind of like damming up water, that I'm storing the water, and then all at once I'm going to release that. Well, I'm going to be damming up hydrogen ions with the energy I'm releasing from what I've been filling my pockets with. So that's kind of what I want you to keep track of. So we're going to build up those hydrogen ions to run down that concentration gradient. So here's a representation of that. So we want to know, I want you to keep track of, well, where are we? Well, ever since we left glycolysis, we're in the mitochondrion. And in the mitochondrion, I have 2 membranes. I have an outer, okay, because we are eukaryotic cells and we have membrane-bound organelles. I have an outer phospholipid bilayer of my mitochondrion, and I have an inner membrane. And that inner membrane is folded. And anytime you see folding, okay, you should automatically think, when stuff's folded, I'm trying to increase surface area. So to maximize the amount of ATP I make, given the number of mitochondria I have, I want that inner membrane to be folded so that these chemical reactions have all the surface area in the world to give me the space that I need to allow them to occur. Okay, so outer membrane surrounds the mitochondria, inner membrane is folded within the mitochondrion. Along that inner membrane, I have a whole bunch of different proteins. Okay, you can learn the names at some point. Okay, I have a whole bunch of transmembrane proteins. And what I'm going to be doing is I'm going to take the electrons and I'm going to release the energy I had to place those electrons and hydrogen ions. And I'm going to release that energy, and I'm going to go through a series of oxidation reduction reactions. I'm going to lose electrons only to be gained. And I'm going to lose electrons only to be gained. And I'm going to lose electrons only to be gained. And so I'm going to go through a series of oxidation reduction reactions. And through that process, it releases the energy to pump hydrogen ions. And where are they being pumped? They're being pumped between the outer membrane and the inner membrane of the mitochondria. And it's going to build up, I guess I could ask the question, is this going to build up a concentration gradient? Absolutely, because look what I'm damming on this side, the dam being the inner membrane and the outer membrane, the dams, I guess, keeping it from going out, keeping it in. I'm accumulating these hydrogen ions in that inner membrane, and so I'm producing a concentration gradient, and because they're positively charged, I'm producing an electrical gradient as well. So what's going to happen is I'm going to open a channel, and I'm going to open the channel, and it just happens to be an enzyme, and this enzyme is called ATP synthase. Okay, that should be simple. I have an enzyme that's going to, enzyme because it ends in ase, and I have an enzyme that's going to synthesize ATP. And so as the hydrogen ions run down their gradient, they're going to release the energy to take ADP, put a phosphate on it, phosphorylate it to produce ATP. And it just so happens that for each NADH, now we're doing a little rounding here, don't panic, but it's going to make it simpler. For each NADH I put through this system, I'm going to get 3 ATP. For each FADH2, because it joins a little later in the process, I'm going to get 2 ATP. So now, if you can imagine NADH, okay, if I get 3 for every one I had, well, my pockets were full of that. Because I formed a couple in glycolysis, I formed a couple when I formed acetyl-CoA, I formed 6 through Krebs cycle. And for each one of those, okay, I'm going to get 3 ATP out of it. And for each FADH2, and I got 2 of them in Krebs cycle, because I went through the process twice, I get 2 in that process. Okay? So that's where I'm going to get those from. Now, the question is, well, where did that range? Remember, sometimes it's 36, sometimes it's 38. What it goes back to is, remember glycolysis occurred in the cytoplasm, and we got a couple of NADHs in that process? Well, different cells shuttle, that's the term we use, shuttle those NADHs into the mitochondria differently. And depending on that difference, depends on did we get 2 ATP out of them or 3 ATP out of them. And that's why we get that range of, in some cells, 36 per glucose molecule. In some cells, 38 per glucose molecule. Just depends on the cell, depends on that shuttling system. Okay? So that's how it runs down that gradient and produces that ATP. Now, the issue becomes, well, now out here we have these electrons and we have the hydrogen ions down here within that inner membrane. Well, now what are we going to do with those things? Well, the advantage here, and this is where oxygen comes in, oxygen, okay, so let me make this statement first. Oxygen is the proton and electron acceptor in the electron transport system. Let me repeat that. Oxygen's role in this process is to accept the protons and the electrons of the electron transport system. So if I have oxygen, O2, and I pick up hydrogen ions and electrons, what do I get? Yes, I do. I get oxygens plus hydrogen ions plus electrons to make it neutral. I get water. So where does that water come from in this process? It comes from oxygen being the hydrogen ion and the electron acceptor of electron transport. That's what oxygen does. You're like, really? That's it? We've gone through this whole process and talked about how important oxygen is. Well, the problem is if oxygen isn't there, then we basically overload the number of hydrogen ions and we shut the system down way back before Krebs cycle, back at the end of glycolysis, and lactic acid is that product. So if it's not here, if oxygen isn't here, it shuts that process down. So again, NADHs, FADH2s bring the hydrogen atoms, okay, the ions and the electrons, together they're atoms, okay? And we play hot potato with the electrons, we pump the hydrogen ions, we build up the concentration gradient. That concentration gradient runs down its gradient, okay, to run basically a turbine system to synthesize the ATP. So your book does a pretty good job of showing you kind of the entire process, that we're going to pass electrons from this protein to this protein to this protein, but they have also summarized it quite well as well. So kind of using that turbine analogy, that here we've backed up the water behind the dam, we have potential energy. Well, we backed up hydrogen ions behind the dam. We activated the floodgates, we ran the turbine, which then created the energy. So that's what we're doing here. We're taking and activating the enzyme ATP synthase to be able to run the hydrogen ions down their concentration gradient and synthesize ATP. That's what's taking place. And so it's basically a Symport system, because we are pumping everything in the same direction as we release the energy to produce or put together the ADP and P to get ATP. The important part of this, of course, being the role of oxygen at the end. All right, so if we look at the overview of this, and this is what you want to be able to do, is take this graphic or one like it and be able to break it down into, first, our 4 steps. Glycolysis, formation of acetyl coenzyme A, Krebs cycle, electron transport. Find where each of those is located, where each of those is taking place. Take each one individually. Say, okay, glycolysis is, okay, breaking of glucose and forming 2 pyruvic acid molecules. And in that process, getting 2 net ATP plus 2 NADHs. Okay, so let's make sure we look at the graphic. If we don't have oxygen available, all we get is the 2 net ATP, and our product is lactic acid. With oxygen, the process continues. We transport the 2 pyruvic acids into the mitochondria. We strip off a carbon and 2 oxygens off of pyruvic acid, and we get our 2-carbon acetyl group. It then combines with our vitamin, our coenzyme A, which picks up that acetyl group, that's where the A comes from, drops it into Krebs cycle. Krebs cycle, we produce per time through, we produce 2 CO2s, we get 3 NADHs, we get 1 FADH2 and 1 ATP. Okay? Because we get to go through it twice, we get 4 of these and 6 of these and 2 of these. All of our NADHs, FADH2s then transfer their hydrogens and electrons to the electron transport chain, where we get our biggest yield of ATP. How much? For each NADH, we get 3. For each FADH2, we get 2. Okay. Oxygen then picks up the hydrogen ions and electrons to form water. And because that water was produced as part of metabolism, okay, we're going to refer to this in later units as well, because it was produced as part of metabolism, what do we call it? We call it metabolic water. The question is, well, how much of that stuff do we produce per day? Okay, we produce about 200 milliliters of water that you didn't drink or you didn't consume as part of your food, that you then are responsible to excrete, whether you excrete it as sweat, whether you excrete it as humidified air, whether you excrete it into your gastrointestinal system, whether you excrete it through your renal system. But you make about 200 milliliters of metabolic water, where oxygen picked up those hydrogen ions, okay, and those electrons in that process. So if we do the accounting process for our ATP, we got 2 net from glycolysis in the formation of acetyl-CoA through Krebs cycle. Okay, we got 2 more, and then we got our biggest yield, okay, as we went through electron transport, is where we got that from.